My name is Danielle Doucet, and I did my project on the KYASANUR forest disease. Here's a short overview about what we're going to be looking at in my presentation. First, the disease, then history, clinical features, diagnosis and treatment, epidemiology and life cycle, vectors and transmission, treatment, prevention, and control. Here's a little bit about the disease. KYASANUR forest disease, or KSD, is caused by the KYASANUR forest disease virus, or KSDV. Throughout the presentation, I will be referring to KYASANUR forest disease as KSD. It is a member of the family Flaviviridae and is found in Karnataka State, India. So who's at risk for this disease? People with frequent exposure to the outdoors who are more likely to come in contact with ticks. Recently, though, KSD was discovered in Saudi Arabia. So here's a short history of KSD. Deaths in 2 monkey populations led to the discovery of KSD. Illness and some deaths among villagers followed the deaths in the monkey populations. This may have been what prompted the villagers to do research on the disease in the first place. The disease is named after the forest area it was first discovered in, and it was discovered in 1957 in the KYASANUR forest in the Karnataka State, India. Symptoms of KSD include the sudden onset of chills, frontal headache, and high fever, which would be described as about 40 degrees Celsius. So clinical symptoms are a fever for 12 days or longer, severe myalgia, which is defined as muscle pain, cough, diarrhea, vomiting, and photophobia, which is eye discomfort in bright light. While this disease is not usually fatal, there are still approximately 400 to 500 cases per year with a fatality rate of 3 to 5%. The incubation period for KSD is about 2 to 7 days. And often, during a relapse, which lasts about 2 to 12 days, an individual would experience the same symptoms. Additional complications during a relapse include neck stiffness, mental disturbance, giddiness, abnormality of reflexes, and signs of encephalitis, which is an inflammation of the brain. Diagnosis of KSD is generally based on symptoms. The laboratory tests that can be done to test for KSD include hemagglutination inhibition and immunofluorescence. While these are very complicated laboratory tests, here is a shortened version of their definitions. So for hemagglutination inhibition, it is a method of quantification for viruses or bacteria by hemagglutination, which is an agglutination, or a clumping of particles, involving red blood cells. Immunofluorescence is a technique used for light microscopy used on microbiological samples, uses the specificity of antibodies to their antigen to target fluorescent dyes to specific targets within a cell. This test visualizes the distribution of target or studied molecule in the sample. So here's a short life cycle for the ticks that spread KSD. Tick activity is seasonal. The adults become active for a few monsoons at the beginning of monsoon season in June. Their population peak is in July and August and then declines in September at the end of monsoon season. Females lay their eggs and the larvae feed on small animals. Larvae populations build up during the monsoon season but are dormant until forest litter dries up post-monsoon season. Nymph activity is high from January to May, and the nymph activity is associated with the KSD epidemic. This means the nymphs are the important life stage for human transmission. So who is affected? The main hosts are small rodents. Shrews, monkeys, and bats may also carry it. Humans are the dead end of the virus, so after us, it doesn't go anywhere. Humans would catch the virus from a tick bite or contact with an infected animal, like a sick or recently dead monkey. Larger animals can also be effective, like goats, cows, or sheep, but they don't transmit the disease. For treatment, prevention, and control, treating the forest floor once or twice with insecticide is very effective for killing ticks that carry the virus. This is a very difficult way of treating for the virus because ticks are very widespread. Tick repellents, like DEET, DMP, or DDP, are 90 to 100% effective for deterring ticks. This should be used in combination with protective clothing. While there is no vaccine for KSD, vaccines are currently being tested and developed. Thank you very much for listening, and I hope you have a great summer. Please try to avoid hard ticks.